There's a lot of things being currently developed, and automation plays a very, very, very big role into that. What I can see is that a lot now are moving towards stable producer cell lines, something similar to what you have with the monoclonal antibodies. So, with targeted integration of your genes and basically inducing at a certain stage to produce your AAV and maximizing the output of these cells. You need to solve the cell toxicity that happens once you start to activate your AAV production machinery. But if that is being solved, which a lot of effort is going into that right now, you can more automate process and make it in a more continuous manner to increase the efficiency and the production from what you get.
Hey, smart biotech scientists, welcome back. We are in the middle of a conversation with Ahmed Youssef on the development of AAV-based gene therapies. Ahmed Youssef is a senior manager in the process development team at Ascent, an end-to-end gene therapy development partner. What did you think about part 1 of our conversation? Please tell us by leaving a review on Apple Podcast or whatever platform you found us on. And if you haven't listened to part 1 yet, please go back and listen to part 1 first. And now we are continuing our conversation about the development of AAV gene therapies. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics at market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Now, let's assume that we have done our homework. We have now a robust process. We have the right quality. We have a good yield. Now, the next step is to characterize this process. So, what are the different methods or techniques we use to characterize our process?
So normally when you reach that, from a process perspective, you can do that from a process course and analytics to characterize the product. I will not go into the product characterization part. This is not really my area of expertise, but there is a wide range of assays that you can do, post-translational modifications, a lot of things that you can do in addition to the potency and all of these impurity assays to characterize your product. The process itself, at one stage when you need to achieve commercialization, you need to start doing your process performance qualification runs, your PPQ runs. And these where basically you check for process robustness, where you say, okay, my process is robust. It's not a one-off where I got a very good yield, a very good quality. No, every time I do the process, it succeeds, it delivers what's requested, and it falls within the range of the process. In order to do that, to prepare to do that, because you normally do the PPQ runs on the final manufacturing GMP scale, it's very expensive. It also takes time and money. So you try to validate your process on a smaller scale first. So a good thing is always to have a scale down model of your process that you can do the process robustness and characterization on that, understand what influence your process from a parameters, whether it's agitation, whether it's feeding with a certain feed, whether it's a certain nutrient, understanding all that about your process, ensuring that your process is robust enough on a smaller scale would then translate that when you start doing the PPQ runs on a larger scale in a GMP environment, you have the highest chances of success. So you don't need to repeat that. You don't need to waste any resources in that.
From your experience, Ahmed, what is the best small scale, let's say like that, the volume to do these runs?
Yeah, that's a good question. So it depends on the resources that you have and the output that you want to get. Do you want to get high throughput or do you want to just get a small scale? I worked with a lot of suppliers from bioreactors with various modalities, whether single-use bioreactors or reusable, autoclavable, multi-use glass bioreactors. I would say if you're doing cell cloning, for example, or plasmid ratio optimizations, you need the high throughput. Then you would look for a system that has 24, 48 parallel bioreactors, something similar to the Amber 15, for example, from Sartorius. This is the smallest automated controlled bioreactor format I've seen so far, that you can run 48 experiments. You will not be having that to be scalable. That's even highlighted by Sartorius, but it is really indicative in terms of comparison between the 48 conditions to pick the best producer clone. Normally, you start, for example, with cloning with a 96 well plate or 384 well plate. You do your single cell cloning, you pick up the colonies, you narrow that down, and then you need to functionally test that in an actual transfection experiment. Is these clones okay? They are growing very well. They are single clones, but are they producing as well? Very good or not? Are they having the good quality or not? Then you need to narrow down that and do that in an automated format. Some people use shake flasks, very small shake flasks, but the way I personally see it, it's still an uncontrolled condition. You're just shaking, you control the temperature, you control the steering or by shaking, and then you just have the humidity and the aeration in the incubator. But to really have some sort of a pH and oxygen control and having a controlled process, you would start from something similar to the Amber 15. You narrow down your condition or narrow down your clone, you do your DOE, which is very helpful at that stage, and then it depends on how you want to scale it up further. Then you need to have another step, whether a 2-liter bioreactor, an Amber 250 modular from Sartorius as well, which is really helpful because it's, for us at least, that's the starting scaling point. And it really scales up nicely with the whole range of the Sartorius bioreactors. It makes things smoothly, and you have 12 parallel bioreactor units single use. So you can then take from the very, very small scale the best conditions, validate that, and confirm that on the Amber 250, and from that, off you go. If it depends on the final scale then that you want, the capabilities that you have, how you want to transfer, and exactly, do you want to generate more data in between, or do you want to just go to the final scale right away? In my lab, for example, we have from the Amber 15 till the 200-liter bioreactor, every single scale. It depends now. When we get a client that just needs some material at a 5-liter scale, it doesn't make sense to charge them for a 50-liter scale production, so we just do a 5-liter bioreactor. If they want a 10-liter or if they want a 200-liter, so you go directly to the scale. Our starting point is always the Amber 250 because it scales up very nicely, representative volume, representative control strategy, and then you scale it up directly to whatever scale that you want. But that is because, David, the effort that we spend into characterizing every bioreactor scale, building the relation, building the operational conditions between everyone, so then we can jump between these scales very easily.
Yeah, that's excellent. You made a very good point here, Ahmed. You put a lot of effort into characterizing your equipments, and you know exactly what happens at each scale. But let's dive into that a bit more, because obviously scale up is quite an endeavor. It's quite a challenge. What are the typical challenges you face when scaling up, and how do you overcome these challenges, especially whenever you go into larger volumes, like let's say 100 liter, 200 liter, or even bigger?
True. So I would say one thing that I try to keep in mind is you need to identify your key process parameters. And that you try to keep constant across the process, aside from the bioreactor itself. So just from a process itself, like the oxygenation set point, the gassing, we mentioned now we do transient transfection. How do you do the transfection between small scale to the 200-liter scale? Because that would impact the complex formation between your DNA and the transfection reagent, and also would impact at the end your transfection efficiency. So you need to make sure, saying, okay, I do the transfection in smaller scale like that, and I keep using it like that in larger scale. I always tell my team early on, when we are optimizing in the process or developing, always keep in mind, is that going to be transferable or translated in larger scale or not? If not, then what's the point? Why shall I add this chemical in this particular way on a smaller scale if I cannot really do that for technical reasons? If I'm adding a chemical at a very, very high speed in a small scale, which is okay because you have pumps that can do that. But if you want to linearly have the same speed in a 200-liter scale, you don't have enough pumps to do that. For example, just a simple example. So we need always to think when you are designing a process, designing a bioreactor process, keep the scale in mind. Keep always how you will implement that in a GMP setting in the final scale that you have. Because some processes die at the end. They are very, very good at smaller scale. You scale it up, you cannot reach the power input, you cannot reach the steering in the bioreactor. You have mechanical limitations that you cannot overcome. So you need to keep that in mind when you do that in a smaller scale. With that being said, yes, scale up is really challenging. You need also to have characterization data of your bioreactor. You need to understand that. That's why the Sartorius, with their history and with their development expertise in their bioreactors, they have the full data for all their scales. They have in their bioreactors have the same width to height ratio. So geometrical wise, they are scalable and they are in line and aligned together. And that's what makes scaling up with them easy. They have a lot of tools where they can also help you. You need to find what is the most also, are your cells sensitive to shear stress? Then you really need to take care of the tip speed when you're scaling up from one scale to the other. No, my cells, I do a process intensification, so I really need the high KLA, the high oxygen demand that the cells want. So you need to keep that as your primary when you scale it up. Or my cells are growing in a smaller range, so they are not really shear sensitive, and at the same time they're not really oxygen demanding. Then you use the standard where people scale up by power input per volume, for example, from one scale to the other. So different scaling strategies, you will not be able, most likely not be able to make them all linearly scaled up, but you want to avoid to limit any of the scale up conditions from one scale to the other. And now there's even more and more process analytical technologies available, like Raman spectroscopy, getting more and more mobile, getting more and more portable, and with sensors that you just plug in into your bioreactors and understand more about your process with online monitoring. CO2 now, more focus has been put into it. We know that CO2 play a very, very critical role in regulating production and the cell growth as well with the CHO cell lines. Also in the AAV, we're now implementing that to see how that impacts with the balance between CO2 stripping from your bioreactor and the degassing, or the CO2 accumulation inside and the impact on the quality and the yields. So all of these tools come into play when you design the process, when you build the process from one scale to the other. You need to consider them, build the model on a smaller scale with larger scale in mind, and see how that model develops while you're moving up or scaling up your process.
And to what extent do you use models, let's say mechanistic models or hybrid models, and also computational fluid dynamics?
Honestly, I have not used computational fluid dynamics so far. So I have relied on the modeling from Sartorius for that, for their bioreactors, but I did not do that because we haven't faced a serious problem in scaling up so far. So with the normal standard, let's say, bioreactor characterization tools like the power input, mixing time, KLA, we were able to scale our process very nicely so far. What we might be using in terms of models is modeling for, as I mentioned, PAT processes, modeling for Raman spectroscopy using process analytical technologies. So like Raman spectroscopy that we model, and then based on that model, we can then rely on the data that you get rather than taking at-line or offline samples and analyzing that for glucose, lactate, viable cell densities, and keep a keen eye on the process. Another thing that we do is that with the range of the Ember 250 data that we get with a lot of these parallel bioreactors, we have a huge number of runs that we overlay together and then build a model of how, for example, the pH should look across the whole process, how the DO would look across the whole process, the dissolved oxygen, how the steering is, especially for parameters that are impacted by the culture and variable are not constant. And then whenever we have any new batch or any scale-up batch, you just overlay that on your model, and then you see, do I have a deviation or not? Is the low yield because I see here a spike in the temperature for whatever reason or not? So that's data modeling, and there's a lot of softwares that are also capable of doing that.
Let's talk about the regulatory aspects and what are the specifics with respect to AAV gene therapy products. How do you need to navigate that?
We have a very good comprehensive analytical development team. So they are keeping an eye on that. So whenever FDA or EMA produces any new guidelines for the AAV production or manufacture or something related to specifications, or also from when we communicate with them variously and then they say, okay, please take care because you have that impurities level, your specification is very high, so please lower that a little bit. Or your potency, you specify that your potency from the reference is plus or minus 50%. No, narrow that down a little bit. So early communications with the regulators, in addition to seeking that advice and keeping an eye on every basically new set of guidelines that they produce, gives you a better picture. But what you can derive from that is they are getting more and more rigorous and more vigilant into their specifications and keeping a keen eye, because there's a lot of incidences that you see for therapies that fail with patients. So you need to really provide the safest and the most quality, let's say, oriented therapy to the patients. So you don't want the patients to have anything less than the best. So they keep that in front of their eyes when they approve certain medications or when they review the submissions for certain drugs. At the same time, it also, again, depends on the risk-benefit ratio for the patient. If you're talking to a drug or a disease area that there is no other alternatives, so they might be lenient a little bit, depending, especially with also diseases that have low life expectancy and impacting babies, and you have a very quick progression of such a disease, so there is no other alternative. So that might be regarded, and there might be a little bit more flexible with their guidelines respective to these diseases compared to another disease that there is a lot of other treatment options and there is other standard of care. So in this case, they will be more and more vigilant and requiring for the best of the quality for that. It depends from one disease to the other, and it depends on someone really taking an eye and keeping an eye on the regulatory framework and the regulatory guidelines that are being published every now and then.
And what are the common pitfalls or mistakes that we as scientists should be aware of when working with AAV-based gene therapy production?
Such as, so common pitfalls from which direction?
Anything that may fail our program from the quality side or from the process side or any other thing? Like, are there some specifics?
From specifics point of view, I would say quality fails a lot of programs at one point. The lack of the translation between the in vitro data and the in vivo data. So when you say, okay, I have now an AAV molecule, I have it characterized, but then I did my in vivo studies, my small animal studies, they are successful, they're okay. Then I proceed to clinical studies, but then I do not really see the good response in patients. You need to see why you do not see that. What is the problem? What did you miss during characterization? Did you miss, for example, having a potency assay to be able to really understand how efficacious is your drug? Was a different process conducted between the material that was generated for the animal studies was different in the way it was manufactured compared to the material generated when you started to dose patients? Was there any significance? Did you, for example, some therapies, they go from an adherent cell culture to go to suspension. So they do the adherent one first because it's quick and easy, but then they want to go to the suspension one when they start mass production. And then there's different qualities. Is your comparability package that you submitted covering all of these aspects? Do you understand your process to know why this is happening clinically or not? So all of these aspects, I would say, are challenges and could be pitfalls that could basically make any drug or process non-viable for commercialization and then hits these CMC stumbling blocks.
Now, looking forward to the future, Ahmed, what innovations or breakthroughs do you foresee that will make the therapies more efficacious and also the process develop more efficient?
There's a lot of things being currently developed, and automation plays a very, very, very big role into that. What I can see is that a lot now are moving towards stable producer cell lines, something similar to what you have with the monoclonal antibodies. So with targeted integration of your genes and basically inducing at a certain stage to produce your AAV and maximizing the output of these cells. And with that, once that is better established, then I would assume you would then plug in continuous DSP manufacturing, which is not there on the radar, but at one point it will be coming similar when you do that with monoclonal antibodies. You need to solve the toxicity that is being I mean, the cell toxicity that happens once you start to activate your AAV production machinery. But if that is being solved, which a lot of effort is going into that right now, you can more automate, let's say, the process and make it in a more continuous manner to increase the efficiency and the production from what you get. That's why I'm saying capacity is not an issue anymore, because you hear about process intensification. So, I mean, all the technologies that are currently applied and have been used for years in the monoclonal antibody space are being now applied and tested or adapted, let's put it like that, to be fitting into the AAV space as well, to maximize the yield, to maximize the amounts that you get, to decrease the process time. So you bring down the cost of the production of AAV down, because the price of these medications is really, really high. And you want to make sure that you make it accessible to patients. So you need to bring down the cost, making processes that are more efficient. And this is what technology developers are working on in the background.
We have covered a lot of ground today, Ahmed. What is your 20-second takeaway of our conversation?
So I would say know your final process scale, know your final quality that you want to get. So define your TPP early on. Make sure that you abide by your TPP at every scale of the process, and reach out to regulatories early on once you have some sort of a solid process for advice to make sure that you are on the right track. I think if you're a startup, don't try to invest into having all the capabilities. You don't need to invest your money, your seed money, your funding into building laboratories, building the CMC capabilities. There are a lot of companies out there that can do that for you. And then you would focus into the results and focus into the development of your therapeutic, rather than draining all your money into capabilities that you might or might not use at a later stage. Once, of course, you're on a solid ground and things are progressing, start building your capabilities. But there are a lot of capabilities out there that can support you and give you the advice early on to make your therapeutic viable at the end.
Wow, that's excellent. What a great summary. And before we wrap up, I have another question for you, Ahmed, out of curiosity. If you had another person from the AAV space, the opportunity to ask them a question, what question would you ask that person?
That's a tricky one. I mean, there is a lot of people that really, really define that space and worked with that. I don't have someone exactly in mind to ask them a question, but I would be really interested with a lot of people to talk to and try to pick their brains about how did they reach what they reached in terms of, thinking outside the box in terms of a lot of technologies that we have here, like, for example, replacing the plasmids with the doggy bone DNA, for example, to decrease impurities. The work by a lot of CDMOs and a lot of innovators, I would say, in the AAV field is really impressive.
Excellent. Ahmed, where can people connect with you?
I'm available on LinkedIn. I'm a very responsive person by nature. So whether on LinkedIn, just people, if they want, reach out to me and send me a message. I'm more than happy to help provide any support if needed and to give advice to our smart biotech scientists. So I'm more than happy to feel free to drop me a line on my LinkedIn profile and I will reach out to you right away.
Fantastic. Yeah, smart biotech scientists, please use this opportunity, reach out to Ahmed. And Ahmed, thank you very much for being on the show today and sharing all these amazing insights.
Thanks a lot, David. It was really great to have that opportunity, and thanks for the invite.
Hey, smart biotech scientists, thank you very much for tuning in. Have you already left a review? If not, please stop now and hop over to Apple Podcasts or whatever platform you found this on, and please leave a review. It will help us reach more scientists like you and, more importantly, consider topics that are of interest. And perhaps you are developing novel therapies yourself, but you're not so sure how to navigate all the challenges related to the CMC development part. Well, we have developed a fractional CTO service to help you do exactly that. We will help you develop your therapies better, faster, and cheaper. If this sounds of interest, just book a free call with us using the link down in the show notes, and we'll explain you how this works. And we will also analyze your specific requirements and needs, and will help you get started whether this service is of interest and a good fit or not. But we want to help you excel the development of your therapy. So I look forward to seeing you on the call, and I'll talk to you soon. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.